Hello my dear students. Today I am going to discuss about basics of immunology. So when it comes to immunology, for majority it appears as a boring topic. So we'll try to make it as much interesting as possible. First of all, as we all know, immunology is nothing but the defense system of our body, right? So it is the defense system. But when we think of immunology, there are numerous things. If you observe the first half of the picture, there are something called as complement system, some cytokines like TNF alpha, numerous cells, WBCs, lymphocytes, B lymphocyte, T lymphocyte, CD4, etc. So for many of us, it appears like a soup of all these alphabets, right? CD4, T cell, B cell, NK cell, etc. But actually, don't treat it as a soup of these alphabets. Instead, consider it as a multi-layer defense system of our body. Okay. So what is this immune system? That is our defense system basically is made up of innate and adaptive immunity. This you might be knowing. Innate immunity is nothing but first responders. It is the one which will respond first. Adaptive immunity is considered as special forces. Okay. See, innate immunity you can just imagine in olden days, like older movies, whenever they used to portray soldiers. So the same thing, olden movie forts we can imagine. Soldiers will be doing their job at the borders, right? So it is just like innate immunity in the sense they will be responding immediately to the foreign invaders, right? So the response speed will be immediate. Immediate in the sense within minutes to hours, right? But the response to the invasion by foreign objects or materials will be non-specific. In the sense, these soldiers are trained to recognize only some patterns, that's it. They are not much specific. This we will understand it later. And one more thing is, there is no memory in this type of immunity. In the sense, they will not remember the antigens. So there is no much memory involved here. For example, we can see that skin and mucous membrane is one of the strongest component of innate immunity. Similarly, some cells like neutrophils, monocytes, NK cells, right? And some soluble factors such as cytokines and complement systems. So these all will come in the section of innate immunity. Whereas adaptive immunity is like special forces, as we mentioned. So they are very well trained, they know their job properly. So this will be highly specific and they will be having strong memory. So they will remember which type of antigen has invaded, etc. But the thing is, it will be a slow initial response. Though it will be slow, it will be strong, right? In the sense, they will take days to weeks to start their actual action. So in this comes B and T lymphocytes mainly. So this is the difference which we have to know at the basic level between innate and adaptive immunity. So with this basic scenes you have understood now. We have imagined innate immunity as olden day movie fort soldiers protecting their territory from the invaders. Similarly, we have imagined adaptive immunity as finely trained special forces. So we will begin this movie. It is like a spy movie. Already everywhere it is spy time now. You might be seeing the aura of Durandar. So we will also try to cover this topic in the same manner. Now, first point is the invasion by foreign material. That is nothing but antigen. So they are trying to invade our body. How our immune system will react to that. This is the overall thing here. See, for invaders, usually it is like a common pattern. For example, they will be wearing a black color dress or some uniform. They will be covering their face with some scarf or mask, etc. So this is the common picture which we can imagine for invaders. Okay. So what happens? How our body gets to know which is self and which is not self. This is called as pattern recognition. Okay. So there is something called as pathogen associated molecular patterns. Just now we told that these microbes will wear some uniforms that will be remaining same. So if anyone coming in that uniform, our body will recognize it as foreign material. Okay, so this is called as pattern recognition. For example, gram-negative bacteria will have lipopolysaccharides. So this is their uniform. So what our body will do? Whichever microbe coming up with lipopolysaccharide uniform, our body will recognize it as non-self. So what is that our body in the sense? The eyes of innate cells, they will be looking at these invaders. If anyone is seen in these uniforms, that is in that particular pattern, they will immediately notice that. So this is called as pattern recognition receptors. For example, toll-like receptors you might be already aware of. So what these receptors will do? They will be sitting on the surface of our immune cells. So they will be watching any microbe or foreign antigen if invading and it is fitting into the pattern already trained, they will identify it. Okay, so the thing is, they don't eat the bacteria, they will not do anything, they will just identify the microbe. Then they will generate a chemical alarm to start inflammation. So this is their job. This is the job of pattern recognition. Okay, so hope you got the point how our body will differentiate self from non-self. So once these pattern recognition receptors have generated a signal in the body, what happens next? Now WBCs will come into action. So to start the actual fight, we need white blood cells at the site of invasion or injury. So what are those WBCs? First of all comes the neutrophils, right? So neutrophil is called as the first responder. Wherever is the problem, it will arrive first. It will try to fight hard as much as possible and it will die first. We say within 48 hours, its action will be done. And this is the reason for pus formation, right? This is about neutrophils. It is ready to fight and ready to die. Then comes the monocytes. They are nothing but the heavy hitters. They will be long lived. They will not die that easily. And they can eat multiple pathogens, right? And whenever the same monocytes enter some tissues, they will be called as macrophages. Tissue macrophages. You might have heard about some examples like dendritic cells. Dendritic cells, where do you see that? In the skin. Similarly, Kupffer cells, right? In the liver, like that. At particular tissues, they have different names, but basically they are nothing but monocytes. This is another type of white blood cell which will come for the rescue. Now, some special thing about monocyte is they will not only eat multiple pathogens at a time, whatever they have eaten, they will take some pieces of that particular thing to T cells. So they will again go back and present some part of the pathogen to T cells. They will tell that, see, this tastier thing has invaded the body. You also come and enjoy. That's why they are also called as antigen presenting cells. So they are presenting the part of antigen to T cells. This is the speciality. Other type of white blood cell is nothing but eosinophils. They are particularly trained to act against parasites. So they are mainly anti-parasitic. And they release a compound called as major basic protein, MBP. Here the name has some mystery in it. Though there is basic in the name, this compound is toxic acid. This acid can burn the tough skin of helminths. You can see the helminths here, helminth. Their skin will be tougher. So these eosinophils will release MBP and they will burn out the skin of helminth and they will kill the helminth. This is how they will fight. So their job is very clear. Then comes basophils. So basophils are like suicide bombers. Okay, in the sense they are loaded with some explosive material inside them. So that is nothing but histamine. So when they degranulate it, there will be release of histamine causing sudden vasodilation. It can also lead to hypotension sometimes. Hypotension. So this type mainly acts in case of allergies or anaphylaxis. So you can correlate basophils with histamine loaded suicide bomber. Okay. So these are the WBCs which are allocated particular actions. So they will try to come for rescue. Then the thing is, as the WBCs are reaching the border, they are not only trying to fight against the pathogen, they are also giving some signals. They are giving some signals so that entire body has to know that there is some problem. Okay. So this signal message is nothing but cytokines. For example, this is like a coded language, encrypted communication network of our immune cells. Like you can see in the movies, right? They will send a chit. In that, some code words will be written, like Kala Katta Aa Gaya, just for example. So this is the language of immunity. So now these are WBCs fighting at border with the pathogen. Meanwhile, they are sending some signal in the body. That signal is nothing but cytokines. So they are releasing cytokines. Now we will see something about these signals like cytokines. There is something called as pro-inflammatory cytokines. Pro-inflammatory. So what they will do, they are the fire starters. They will start inflammation in the body. One example is interleukin 1. It is responsible for fever, we will say. That is nothing but it will go to hypothalamus and tell to reset the thermostat so that our body temperature will increase. So what it is doing overall? It is increasing the temperature because some pathogens are not comfortable at this higher temperature. So it is trying to again fight in its own way. So that is leading to fever, right? Then comes interleukin 6. This interleukin will go to liver and stimulate liver cells to produce acute phase reactants. One of the example is C-reactive protein, right? Then one more pro-inflammatory cytokine is TNF alpha. This is called as master regulator. In the sense, if it is released in small quantity or small amount, it will help in fighting against pathogen. But suppose its quantity is large, then it can lead to septic shock also. So we have to be careful with this particular chemical. Similarly, there are some antiviral cytokines. For example, interferon alpha and beta. Interferon alpha and beta. They are mainly produced by virus infected cells. So they are mainly produced by virus infected cells and they will inform the neighboring cells to lock the door. They are like, whenever a cell is infected by a virus, it will give or generate some signals, that is interferon alpha and beta. These signals will go and tell the neighboring cell that there is some problem. So you be safe. Just close the door so that the virus will not attack you next. This is the action of interferon alpha and beta. So overall it will help in stopping the viral replication, right? Similarly, there is something called as interferon gamma. It is mainly produced by T cells and NK cells. And this cytokine is like elite killing machine. Again, helping in fighting pathogens very strongly. Then there are some growth and recruitment cytokines. For example, interleukin 2. 2 in the sense, it will tell the T cells to become 2 from 1. That is nothing but T cell proliferation. Interleukin 8. 8 for recruit. So it will help in recruiting more neutrophils to the site of war. Overall, again, this will also lead to pus formation. Then there are some cytokines which are anti-inflammatory. They are like fire extinguisher. Commonest examples are interleukin 10 and TGF beta. So they will dampen our immune response once the pathogen is cleared. Enemy is killed now, so no need to fight further. That's why these signals will indicate body that stop the fight now, otherwise it can harm our own tissues. This is the action of anti-inflammatory cytokines which will come into action once the pathogen is cleared. These are some signal molecules, cytokines in our body. So in the beginning, we told that in innate immunity, there are some cells like neutrophils, monocytes, and there are some pattern recognition molecules. We spoke about cytokines in soluble molecules. One more pending thing is complement system. So what it is doing? Why is it named like complement system? Whom is it complementing? They are nothing but the cascade of proteins that will complement the actions of antibodies. That's why they are named like complement system. Mainly their actions are remembered using 3 O's. First O is opsonization. So mainly a complement molecule called as C3b. So what it will do? For any pathogen, suppose if you consider this bread as pathogen, so this C3b will apply butter to it, so makes it more tastier, so that other antibodies or other immune cells find it more tastier, so that they will completely eat that pathogen. So this is nothing but buttering of bread, just can be comparable with opsonization. Then there is something called as organization. Mainly complement molecule C5a. It will recruit numerous immune cells. So it will organize everything at the site of need. Then there is something called as osmotic lysis. Again, a component of complement system called as membrane attack complex. So it is a complex which will attack the pathogen, like it will form pores through the membrane and it will attack the pathogen completely. So these 3 actions are very much peculiar about complement system. So overall it is complementing to the other aspects of immunity, right? Okay, now we have done with the innate immunity. So we will take a break. So till now we have discussed about how pattern is recognized. Anyone coming in the uniform, typical pattern with mask and all, they will be recognized. At that site, WBCs will come for fight and they will generate some signals of cytokine, so that body will come to know that there is some problem and complement system is doing its action, right? But in this break, you just think how WBCs got to know that there are some invaders at the border. How did they reach the border? You know that WBCs are there in the bloodstream, right? So how did they reach there? They didn't get any signal. So we will see after the intermission. Okay, after the intermission, as I told, we will see how WBCs have reached the site of problem. We know that WBCs are running fast in the bloodstream, right? So this is bloodstream. We can now see they will be running fast inside the blood vessel. Suppose there is some injury at one particular site of our body. So there, the blood vessel, whatever is there, will create some humps. So they will try to slow down the speed of WBCs. So these humps are created. What are those humps? They are nothing but selectins. Selectins. So now WBCs will roll over the humps. Automatically speed will come down. So this is called as rolling over. And then what happens? The same blood vessel, now the speed of WBCs has slowed down, right? Now it will just attach a velcro so that the WBC will strongly get adhered to the endothelium, right? So that is nothing but integrins. Integrins, like velcro. They will come and attach to the endothelium and stop their movement further. Hope you got the point. Selectins are like humps, like speed breakers. Humps. First it will slow down, roll over and gets adhered strongly. That is nothing but adhesion. Then we told that this is the site of problem, right? So endothelium will have some gaps through which these WBCs will slowly, they will migrate. So this is called as migration or diapedesis or transmigration. Okay, transmigration. You might have seen these cats just migrating below the doors, right? Similarly, WBC will migrate through the gap in the endothelium. Then what they will do? They will be like sniffing. They will take that smell from where the injury has happened. So they will start moving towards that site. This is called as what? Chemotaxis. Chemotaxis. So the WBC which has migrated will slowly move towards the site of injury because of chemokines, that smell. This is called as chemotaxis. That's how they will reach the site of injury. So this is also a question for short answer kind of thing, how WBCs will migrate to the site of injury. So hope you have understood the point. Now we discussed so much about innate immunity, like the old movie Fort Scenes. What these specially trained special forces are doing, that is adaptive immunity, B cells, T cells, that we will see now. We told that innate immunity is doing its action, and furthermore it is helping adaptive immunity in some ways. For example, we told that monocytes, if you remember, they are like hard hitters, we told. So they will eat the pathogens, multiple pathogens, and they will go and present some part of the tasty pathogen to T cells. Okay, this is nothing but antigen presenting cell. This we have discussed. And one more component of innate immunity, that is complement system. Complement system is doing what? It is buttering the pathogen so that it will become more tastier for whom? For the antibodies or adaptive immunity cells, so that they will clear it more efficiently. So this was the action of complement system. So adaptive immunity is getting the help from innate immunity. But the problem is, this adaptive immunity, they are little bit blind, in the sense they cannot find out free floating antigens. If you observe this image carefully, they are blind. Even if the antigen is passing nearby them, they will not be able to recognize it because they are blind to those free floating antigens. So for them, these antigens has to be served in a platter. So they are like, they want mehman nawazi. So they must have the food or the antigen served to them in platter, just like this, by someone. Then they will start their action. So what is this platter? It is nothing but MHC molecule. MHC molecule. For that we have a rule of 8. That is nothing but these adaptive immunity cells are very particular in the platter also. Like for example, CD4 T cells, T lymphocytes, want MHC 2 platter only. So if you present the antigen in the MHC2 platter, then only these CD4 T cells will come into their action. Similarly, CD8 T cells need the antigen or the food to be served in MHC1 platter. So if you observe here, 4 into 2, 8, 8 into 1, 8. Overall, it should be 8. Like that, they are so particular in the platter selection also. So they are little bit extra demanding, we can say. But once it is presented, then B cells and T cells start their action. If we discuss about B cells, it is also called as humoral immunity. So mainly it will act through antibodies. Suppose this is a B cell or plasma cell, we can consider. It will start producing multiple antibodies in the serum. So they will start fighting against the antigen. That's why this is called as humoral immunity. The thing is, they will mainly target extracellular pathogens. For example, suppose the bacteria is in blood or the lymph, then B cells are very active against that. And we know that these antibodies, there are totally 5 isotypes, like 5 immunoglobulins. We can remember that using that famous mnemonic GAMED. So IgG, IgA, IgM, IgE, IgD immunoglobulins. Then What about T cells? They will lead to cell-mediated immunity, not humoral. It is cell-mediated, like CD4 cells, CD8 cells, like that. So what they will do? They will act mainly through perforins, granzymes, and apoptosis. See, this is very, very like lethal, we can say. Suppose this is a microbe. Perforins, in the sense, these T cells will release some chemicals. They will cause pores first in the membrane of the microbe. So all the fluid will start entering into the body of microbe. It will burst out. Granzymes, they will grind the pathogen completely. Apoptosis, in the sense, these T cells will plan the death of these microbes. So in this multiple lethal manner, it will kill the pathogens. You might have seen this movie and this famous dialogue. What is that? Rahman Dakayat ki di hui maut. Something he will tell in the same manner, no? Like overall, it will be very lethal, similar to T cells action, we can say. And here the point is, they will target intracellular pathogens also. If the pathogen is hiding inside the cell, they will go find out that and they will hit them very hard. For example, viruses inside the cell, they will kill them also. Even against cancer cells, T cells are the one which will fight. So overall, they are very lethal, we can say. And we told that B cells will act through these 5 antibodies. We have to know about them also little bit. G is like the commonest one. It is guardian. Like we can compare this antibody with Bahubali Kattappa. So whatever happens, he is ready to protect any number of generations of that particular family. In the sense, this not only protect us, it will pass through the placenta to the baby, newborn, and it will protect the next generation also. Placenta crossing is seen with immunoglobulin G. Immunoglobulin A is mainly in the secretions like our tear or saliva, we can say, even the breast milk. That's why breastfeeding is also very, very important to get the immunity from mother. IgM is a macromer, like it is a huge pentamer, we can say. Pentamer. Again, it will respond first among all these. This is the first responder. IgE is mainly for allergic, parasitic problems. It will take care. IgD, its action is not that like clear. Its action is not that clear overall. So these points we have to know. Even in the entrance, they will ask which antibodies can cross placenta. It is nothing but G, guardian, Kattappa. Then which is there commonly in the secretions? It is IgA, which is the one which will first respond, IgM. E is mainly for allergy and parasitic. So this is the overall basics of immunology. Hope you have understood it in a very cinematic way. I hope it was helpful for you. Share it among the undergraduates as much as possible so that they will understand the basics of immunology, so that anything learnt above this level will be easier for them to imagine. And also, if you are liking the content in our channel, please subscribe to our channel. It will be a great support to us for coming up with such informative videos. See you all in next video. Thank you so much.